Welcome back, folks. This is going to be Chapter 2 about Mendelian genetics, also known as transmission genetics. The chapter essentially has three parts. The first part looks at Mendel's history and the types of experiments he performed and their implications. The middle part of the chapter looks at some mathematical probabilities. The chi-squared analysis is included. And the final part of the chapter looks at alternative ways in which genetic inheritance is performed depending on which chromosomes the genes reside on. Gregor Mendel is the father of genetics. There's no doubt about that. His history is quite complex. He wasn't a simple person who went into the priesthood. He was, in fact, a scientist, and he wanted to become a teacher, and unfortunately he did not have a chance to complete his examinations in Austria that are necessary to become a fully fledged teacher. So what he did, he decided to enroll in the priesthood as an alternative way in which he could still practice without his teaching credentials. His use of pea plants wasn't coincidental. It just so happened that a lot of people at the time, a lot of scientists, biologists, were working on pea plants. And in the market close to his monastery, these peas were available readily. In total, his notes indicate that he acquired 34 different varieties of peas from these local markets. And he examined the characteristics, or let's call them traits from now on. He examined the traits of each to see if there was something suitable that he could study. And he ended up narrowing down that search to 14 strains representing 7 specific traits, each of which only had 2 forms. And this is where we need to be very thankful that he didn't study something a bit more complex. But he was a scientist and he was very methodical. So those 2 forms of each trait he could easily distinguish. And he worked on these 14 strains for 5 years, doing planned crosses and keeping meticulous notes. If you get a chance to sit down and really study his history, it's a fascinating episode of how meandering in careers can lead to some useful outcomes. Figure 2.1 shows you the 7 traits that he studied, each with 2 forms, or dichotomous. The first was the color of the seed. So his seeds were either going to be yellow or green. The second trait was the shape of the seeds, either wrinkled or round. Then we move on to the pod itself, the container for the seeds. That also had 2 colors, green or yellow, and it also had 2 shapes, referred to as either inflated, which is smooth, or constricted, which is really wrinkled. The flower also had 2 traits. The colors of the flower were either going to be purple or yellow, nothing else. And the position at which the flowers grew on the plant were either at the very ends of the branches or along the branches, axial versus terminal. And finally, he was able to analyze his plants in terms of height. They were either tall or they were short, up to 2 feet and from 6 feet above. The results of his 5-year analysis were published in 1866 in a paper. And he actually did send that paper to Darwin, but Darwin is noted to have not even opened the correspondence. Regardless, it took another 34 years for people to really appreciate what Mendel had done, and during that time he had passed away. In 1900, his work was discovered by 3 independent sets of biologists in Europe, and that rekindled the interest in Mendel and the field of genetics. In addition to his reasons for choosing to work with the pea plant, easy to work with, easily available, and widely studied, he used his training in physics to methodically apply the scientific method to his experiments. And his success stemmed from him doing a quantitative analysis. So counting was the key. And that's why I think he became known, and other scientists who didn't count things, they didn't get credit. At that time in the 19th century, the most popular hypothesis for the way that features were inherited by creatures from their parents was known as the blending theory of inheritance. And in that regard, children were considered to be an intermediate mixture of the juices present in the egg and the sperm from their parents. So under that theory, a black and white cat, if bred together, would produce gray kittens. And the black and white traits would never reappear if the gray kittens were crossed with each other. And of course, it didn't take Mendel very long to disprove that particular component of that old blending theory. This is a great point to now take a pause and review the scientific method and its components. So you may already remember from previous courses that the scientific approach has many categories which should be implemented in the correct order. So the very first thing we do as scientists is to make observations about things that we see, and from those observations we generate a testable hypothesis. And a testable hypothesis is one that can be tested using the tools that we have, and also it can be falsifiable. Once the hypothesis is at hand, then the next thing to do is design some controlled experiments to test that hypothesis. And the appropriate controls must be included in the testing. Once the data is collected from the experiments, it has to be then analyzed. Analysis of the data could result in comparing these to other hypotheses under different conditions. Conclusions are drawn, and from those conclusions, then inferences are made. If one needs to reformulate the hypothesis, that can be done too. The reason that Mendel was successful and others before him were not, in terms of analyzing genetic inheritance, was that his experimental methods were very carefully designed. And he was able to meticulously record everything in his notebooks. So that is something that we should keep in mind. Because his notes are available to us today, we can then dissect those to see what he did in particular that led to his success. And there were 5 criteria that have been identified in his experimental innovations. The first is he conducted controlled crosses. So he, for many years, was able to make sure that he understood the genetic composition of each strain of pea plant that he used. The second thing, he utilized pure breeding strains. A pure breeding strain is something in which the genetic makeup is pure, i.e. big A, and not any mixing of small a. And big A in this case will be the dominant allele, and small a will be the recessive allele of that same gene, gene A. The second, next, his choice of selecting the right type of experiment to perform. In this case, the characteristics that he studied, the traits, they were dichotomous, and he made sure that they were dichotomous, and anything that wasn't, he basically set aside. He then measured his results and then used those measurements to look for patterns. And this is the important thing. The patterns were the major thing that drove Mendel to keep going and conclude his experiments. And to do that, in order to make sure the numbers were reliable, he had to replicate his experiments multiple times, and then he also performed the reverse matings. So he took a male plant that was tall and a female plant that was short, and he mated them, and at the same time he had the reverse, a tall female plant mated with a small male plant. And he performed test crosses, and we need to learn what test crosses are as a geneticist, and we'll be looking at that data very shortly. Unlike most animals, most plants are able to reproduce either with alternative partners or by themselves. And self-fertilization is where the same bush, the same plant, will produce both male and female parts, and they have the opportunity to fertilize themselves. Under normal wild circumstances, pea plants are normally self-fertilizers, but in the presence of agents such as insects, birds, and the like, which can carry that pollen to alternative individual pea plants. In Mendel's case, he didn't want to leave anything to chance, so he meticulously used paintbrushes to artificially cross-fertilize his plants. Figure 2.2 illustrates the life cycle of the pea plant, Pisum sativum. The important thing is that after fertilization between the pollen and the ovule, the egg, the development of the seed is the next generation. So the seeds are the babies. And in this case, we have 7 seeds in this pod, and each one of them has the potential to then germinate and then produce the next generation. Figure 2.3 illustrates the process of artificial cross-fertilization employed by Mendel. He took precautions in removing the anthers from one parent so that there was no possibility that the pollen of those anthers could fertilize the ovules of that same plant. So once he had these manipulated flowers, he was able to then use the pollen from the second parent and then pollinate for sure the ovules of the first parent. And that led to the production of the seeds, and then the seeds were taken out of the pods and germinated to produce the progeny, the F1, the children. Mendel took the time to guarantee that before he started his experiments, that all his biological material had a known genotype, the genetic makeup. To do this, he established in his garden different lines of plants that he bred together again and again until he eliminated the impure characteristics. For instance, in the tall plants, he would breed them together continuously until there were no short plants whatsoever after many, many, many generations. And as it says here, it took him 2 years to establish those lines. Those lines are known as pure breeding or true breeding strains. So with respect to the 7 traits that he studied, he must have had 14 different areas of the garden in which pure strains of these different flowers grew. Each experiment that he performed, he is noted to have begun with crosses between 2 pure breeding parental strains, thus giving him a guarantee of their genetic composition. In genetic analysis of this nature, the parent generation is always designated as P, and their offspring, their children, their babies, as the F1 generation. F stands for filial, and in Latin that means offspring. Students are expected to use these abbreviations when planning out and reporting their genetic crosses. Some of Mendel's genius lay in the way that he planned things. One thing that is obvious is had he stopped investigating at the first F1 generation, then his results would not have revealed anything, because in the crosses that he performed, the F1 generation all had the same phenotype. So either it had yellow seeds or it had tall plants, but he never saw the other characteristic of the other parent. What he did, he took the F1 generation, the children, and he bred them together. And in plants, that's easier to do. And then he looked at the grandchildren, the F2. And that's where the secrets of genetics revealed themselves. In many cases, he proceeded to look at the great-grandchildren, the F3, and so on. So by using these abbreviations, P, F1, F2, F3, one can relay the different generations that are being experimented upon. Figure 2.4 is very important for beginning geneticists because it illustrates exactly the principles that we're trying to learn. And at the same time, the findings that Mendel generated every time he crossed his plants together. The crossing of pure breeding purple plants with pure breeding white plants would be the parental generation, and that cross mating would lead to the production of the children, and that will be the F1. When the children are crossed with themselves, mated with themselves, then it produces the F2, the grandchildren. In all the initial experiments that Mendel performed with pure breeding crosses, he found that the number and types of plants produced in each generation had a consistent pattern. Every time he crossed 2 traits, only 1 trait appeared in the children, the F1. So all the offspring were the same, 100%. When he crossed them together, he found that in the F2, the grandchildren always had a ratio close to 3 to 1. And this is very important. The disappearance of one of the traits and its reappearance in the grandchildren was of particular intrigue to him. Let's be very careful in the use of the word trait. So in the previous few minutes, I've used the word trait to refer to, say, yellow seeds, and another use of the word to refer to green seeds. But that's a mistake in genetics. A trait is a single characteristic that can occur in alternative forms. So I should be referring to seed color as my trait, and then the yellow seeds will be one phenotype, and then the green seeds will be the alternative phenotype for that one trait. As we've mentioned several times, Mendel was careful in filtering down what he studied. So for every trait, he made sure that in that particular strain of pea plant, there were only two possible phenotypes. Not only that, he made sure that they could be easily distinguished in different generations. His meticulous recording of numbers is the key to his success. By counting the number of progeny of each type of plant and then counting the offspring that each cross produced, he was able to identify patterns. And those patterns gave a consistent ratio between the different types of phenotypes for each trait. And these ratios are the most important thing to emerge from Mendel's work. These mathematical patterns are the foundation of the laws of heredity generated by Mendel. Notes reveal that he made the same crosses multiple times again and again, and these are known as replicate crosses. In science, if possible, replication is desired if not required. He is noted to have tested everything, including reciprocal crosses, in which the sexes of the parents are reversed and then their progeny are analyzed. And then finally, he performed test crosses. Now, test crosses are something that we need to learn. A test cross is normally performed between an unknown individual and a known individual. And the known individual always has the recessive form of the phenotype for that particular trait. So in this case, we know that the yellow seeds and the green seeds, when they're mated together in pure form, the green seeds disappear in the F1. So the F1 tells us that the green seeds are recessive, and they would be ideal for then use in a test cross. More about test crosses as we show you data. Within a very short time of Mendel beginning his true experiments, he was able to quickly disprove the blending theory of heredity that was the paramount hypothesis at the time. And he came up with a new theory that we'll look at in a few seconds. So once again, his experiments always started with a crossbreeding experiments of two pure breeding strains of these pea plants to yield the next generation. And then he took those F1 offspring and he bred them together to produce the F2. In every case, he got consistent results. Doesn't matter which trait he studied. When crossing pure breeding parental strains, all the F1 only displayed one of the two phenotypes of one of the parents. The other one had disappeared. For example, when breeding the tall plants with the short plants disappeared and all the F1 were tall. And there's another example here to do with the color of the peas. The green peas all disappeared. In fact, a revisit to figure 2.1 shows us that the top row is the dominant phenotype for each trait, and the bottom row is the recessive phenotype for each trait. Anytime this relationship exists between phenotypes of a single trait, we call that the dominant phenotype. And the one that disappears is always called the recessive phenotype. So please remember the spelling of recessive. In every case, when the F1s were bred together, crossed, the F2 always revealed a 3-to-1 ratio of the two phenotypes, with the recessive phenotype reappearing in the grandchildren. So a 3-to-1 ratio is 75% to 25%. So 25% of the offspring, the grandchildren, would be green seeded, and 3/4, 75%, will be yellow seeded in the example from the preceding slide. A couple of other genetic terms to remember has to do with the way that we refer to the genetic makeup of a individual that carries 2 chromosomes. So pure breeding individuals, as the types that Mendel had established for 2 years, they would have identical copies of the 2 alleles for that particular trait. So alleles are alternative forms of the same gene. Alternative forms of the same gene. An individual that has 2 alleles of the same type, big A for instance, or small a, for instance, would be homozygous. That tells us that the 2 chromosomes are the same, homozygous. If an individual has one big A and one small a, then they have 2 different alleles, and that would be referred to as heterozygous. So in the case of Mendel's crosses, we can put the information that we've learned so far into this relationship here. The letter G refers to that particular trait, in this case, the color of the seed. Because we have 2 alternative forms, we can use the big G to represent the yellow allele and the small g to represent the green allele. So by convention, when you have a pure breeding strain of individual, they do not have a mixture of anything other than the one letter, one allele. So the yellow seeded parents will be big G, and the green seeded parents will be small g. And then through fertilization, their babies, their offspring, would be heterozygous. So these are homozygous pure breeding parents, and the babies are heterozygous. They have the big G and the small g. If you take the F1 and you breed them together, you will end up with the F2 that will have various combinations of these genes. And we're going to study that in more detail later. The important thing is the word segregation. That's the principle behind figure 2.5. Segregation simply means separation. You can see in this example here that the big G and the small g are together in the same nucleus of these cells, but during the formation of gametes, pollen or eggs, the gametes are separated, and that is segregation. Separation means segregation. So Mendel was able to deduce that there was some type of separation going on between the F1 and the F2 with respect to the nuclear content of their DNA. In summary, these initial experiments performed by Mendel always produced the following outcomes. There was always dominance of one phenotype over the other in the F1 generation, the children. In the F2 generation, the recessive phenotype re-emerged. And the ratio in the F2, not the F1, but the F2, the ratio was always close to 3-to-1, dominant to recessive. Table 2.1 shows us this data taken from Mendel's notebooks. And even though the ratio isn't exactly 3-to-1, within mathematical error, it is 3-to-1. Each row of this table represents the 7 traits that Mendel studied. Based on the way that he thought these particles inside cells were being inherited, each particle had a particular nature, big G or small g, he was able to propose the theory of particulate inheritance. And that's the theory that we live with today. In his genius, he was able to deduce that when plants and animals make gametes, the gamete only has space for one of the 2 parental alleles. So if a parent has 2 big G's, then the gamete will only get one of the 2 big G's, and it doesn't matter, it's going to be a big G. However, if the parent is heterozygous and they have a big G and a small g, then the gamete can get either. And that is what is stated in this bullet point at the bottom of this slide. We now call those hereditary particles that he envisioned as alleles. And alleles are nothing more than the alternative forms of the same gene residing on the chromosomes of that particular creature. It is thus the combination of alleles that will then decide the phenotype. When one is studying one gene and ignoring all the other genes in that particular species of organism, we call that a Monohybrid-Cross experiment. So these breeding experiments are just studying one of the many genes that the organism can have. And a cross between two pure breeding monohybrid cross experiments always produces a heterozygous F1. Then when these F1 are crossed together with themselves, that is big G small g and big G small g in each of those two F1s, then in the F2 we get that 3:1 ratio. And that's called the phenotypic ratio, because that's the observable outcome of the genetics, the outward appearance of these pea plants. The genetic ratio is something different. The genetic ratio is the ratio between the two alleles inside each individual. This 1:2:1 ratio simply refers to all the different possibilities that the F2 generation could be. So 1/4 will be big G, big G. 1/4 of the F2 will be small g, small g. And 1/2 will be a combination of big G and small g. Sometimes things can get confusing in genetics, especially if you have too many moving parts. So in order to help geneticists keep everything aligned, Reginald Punnett came up with the Punnett square as an easy way to predict the outcome of the fertilization of sperm and eggs, or pollen and eggs. In the use of this tool, the gametes produced by one parent are placed on top, and the gametes produced by the other parent are placed down one side. And then by bringing the two labels together, you can predict what the genotype of the potential future individual will be, the offspring, and also from their relationship you can predict the phenotype. In this example we have a homozygous recessive green-seeded parent, and we have a heterozygous yellow-seeded parent. The offspring that they'll produce will be predicted by this box, and the number of boxes and their relationship will also give us the ratio. Thus, if this pollen and this egg unite, the pea that will be produced will be heterozygous. And because the big Y is dominant over the small y, its phenotype will be yellow. Likewise here. However, if the small y from this parent and the small y from this parent were to unite, then the offspring will be homozygous recessive, two small y's, and two small y's give rise to a green. Based on these interpretations, Mendel was able to propose his first law. His first law is the law of segregation. And the hypothesis behind the law is described here. And it says that the separation into gametes of these hereditary units, the alleles, and then their random coming together in the gametes of the offspring can be mathematically predicted. And it was applicable to all 7 traits being studied by him. So let's bring back the terminology of the test cross. It's one of the things that geneticists need to learn, and sometimes students misinterpret. What is a test cross? That's the question that the next couple of slides will answer. One unfortunate relationship amongst alleles is that we cannot sometimes tell the difference using the phenotype between an individual that carries big Y, because they'll have yellow seeds, and another individual that carries big Y, small y, because they'll also have yellow seeds. So it's impossible to tell unless you do some other investigation. And that's what a test cross is. It's a way of deciphering the genetic makeup of a person or individual that could be either big Y, or big Y, small y. Now, on the contrary, if you have two small y's, your phenotype will be green. There's no doubt about that. So that one thing is a fact. but we don't know the difference between a big Y and a big Y small y. That's where a test cross comes in. A test cross is simply an individual that will be small y, and have a green seed phenotype. And if you take your unknown and you cross it with this, that will then tell you the answer to your question. And that's what's illustrated here. In this example, let's have a look. So here's our test cross individual. We know that in this case, the small r is the name of the gene. In this case, they're going to be homozygous recessive. We know that. And they'll have a wrinkled seed. So that's our test cross. We're not sure what this individual is. It could be big R, giving rise to the smooth phenotype, or it could be big R, small r, also giving rise to the smooth phenotype. Which one of the two is it? Suppose this was unknown. We would have to find out. And then the answer is, by breeding these two together and looking at the offspring, we can tell. But let's not confuse ourselves right now. Let's begin with this particular cross. So the phenotype of the one parent is big R, and the phenotype of the second parent is small r. When they're crossed together, we end up with this individual here. So Mendel did this cross, and he saw that all the offspring had a smooth seed. But he wasn't sure if that smooth seed was caused. He wasn't sure if that smooth seed surface was produced by a big R combination or big R small r. He wasn't sure of this. So what he did, he took a pure breeding individual and he bred that to the unknown. And what he obtained was a ratio of 1:1 in the resulting progeny. That told him that the genotype of this parent here must have been heterozygous. It could not have been homozygous. If this parent had been big R, then all the babies would have been smooth seeded, containing at least one big R. Thus, even today, we can use test crosses using a homozygous recessive to test for the genotype of a dominant character. That's what a test cross is. Mendel's own data showed that he had heterozygous individuals in the F1 every time. Even though he never saw the recessive phenotype, he knew now that they were heterozygous because he performed a test cross. Mendel predicted using his mathematical training before even the results of these crosses were available. The breeding together of two heterozygous F1 individuals will produce the following gametes in each one of those F1s. The gametes produced by the F1 are indicated for one individual here and the second F1 here. And the mating between this pollen and this egg will give rise to an F2 with a big R genetic genotype and a smooth surface phenotype. In this case, we will get a heterozygous offspring, F2, and it would also have a smooth seed surface. And likewise here. In the last box, we have a small r coming together to produce a homozygous recessive phenotype, and that will be wrinkled. The important thing is, you can't tell the difference between these three boxes from the outside. They all are smooth. But the ratio of them is, for every one of these, there's two of these. For every one of these homozygous dominant, there's two heterozygotes. And that's what we see here, right? The heterozygous state is 2/3 as likely as the homozygous state. We can ignore this box because we know that that's going to be wrinkled. So take that out of the equation. Out of the three remaining boxes, which ones are which genotype if they are smooth? And that's what this is pertaining. The good news is that then and today, when doing experiments like this, there's an agreement between what's expected and what's obtained. The expected was a 3:1 ratio. What he got was very close to a 3:1 ratio every time. Mendel was able to confirm these results by performing a breeding experiment amongst the F2 plants through self-fertilization. and examining the ratios in the F3. And he indeed did get a ratio very close to the predicted in each case. Next, he turned his attention to looking at more sophisticated interactions in terms of the genetics. A dihybrid-cross is nothing more than looking at two genes, two traits at the same time. And a trihybrid-cross will be a genetic experiment looking at three genes at the same time, or three traits. And these experiments led to his second conclusion, his second law, that of independent assortment of alleles. What this means in lay language is that irrespective of which combination of traits he examined, each one was inherited individually in the ratios that we just looked at. Thus, each gene was acting independently of the other genes for the other traits. Let's look through the data for a dihybrid-cross, i.e. two genes. Students must remember to use two different letters of the alphabet, one referring to one gene and the other referring to the other gene. And they must treat each letter independently of the other. In this particular example, we're going to look at one gene referred to by the letter R, either big R or small r, it doesn't matter, that's the same gene. And then right next to it, in the individual, we'll have another gene somewhere else on the chromosomes that we'll call G. And there'll be two varieties of that, big G and small g. When one breeds these two parents together, the gametes that they produce will each only have the opportunity to carry one R and one G. That's very important. Only one R plus one G for every gamete. So in the case of pollen, for this one, all the pollen will be big R, big G. And in the case of the eggs for this parent, again the same thing applies. They can only carry one R and one G. It doesn't matter in this case because they're both the same. So that gamete will carry small r and small g. When these come together in a random fashion, because they're all the same, the offspring, the F1, will be heterozygous for the R locus and heterozygous for the G locus. So double heterozygotes. But when these are bred together, when these make gametes, those gametes will be made in equal ratio, 25%, 25%, 25%, and 25%. And those gametes can then produce the F2. So we're looking at a step in between the F1 and the F2 down here. These will be the gametes of the F1. And that's very important to understand. Figure 2.8 relays the information we just covered as a graphical representation. So here's one parent, pure breeding for two different genes. Here's the other parent, pure breeding for the same two genes, but in the recessive condition. Then they're bred together and gametes are formed, and we'll learn a bit more about this process in a few minutes. And then this will be the combination of gametes that are coming together. The blue have come from this parent, and the red have come from this parent. And when they unite, that's called fertilization, the baby will be then carrying chromosomes that carry this combination. So 2 chromosomes for the R and 2 chromosomes for the G. And the phenotype of the baby will then be an joint interaction between the two. So here, that will produce a smooth, and this will produce a yellow seed. A useful tool for predicting the combinations of gametes is called the forked line diagram. Not my favorite, but it works. So we have one parent here, and it happens to be heterozygous in this example. And then we want to predict all the types of gametes, i.e. sperm, that this individual can produce. So what we do, we deal with one allele at a time. So this gene is dealt with its 2 alleles, and then the second gene is dealt with independently. So looking at the R side first and ignoring the G, there's 2 R's, and they'll be present in the possible gametes in half and half ratio. And then we take each half and then we look at the second gene and we do the same thing. And then we end up with 4 possibilities. And their frequency in the pool of sperm will be 25% for each category. If that is true, then we can put the gametes on top here for one parent in the F1. And we can put the gametes for the second parent in the F1 along the side here. And now, because we have 4 types of sperm and 4 types of egg, we need a bigger Punnett square. And this Punnett square is a 4 by 4 square, simply because of the types of gametes produced. And then all we do is merge the alleles from the sperm with the alleles from the egg into the same box. So big R, big G comes down from here, and big R, big G comes across from here. And the result is this either future or current offspring that contains a homozygous dominant relationship between its 2 genes. You do the same thing across the entire grid and you end up with a pattern. And the pattern is, in terms of genotype, the following. In terms of phenotype, the following. So you're familiar with the phenotype, that 9 to 3 to 3 to 1 relationship. That means 9 of these boxes have the homozygous dominant for each locus. 3 of these boxes have at least one homozygous recessive. 3 of the other boxes have the other as the homozygous recessive. And the very last box, one of the 16, has the double homozygous recessive. One should always get this pattern if you breed together in a dihybrid-cross 2 parents that are both double heterozygotes. Mendel first predicted this ratio, and then he performed the experiments and he got this ratio. So it's very close to what he predicted within experimental error. So his results told him that the mathematics and the outcome from the genetic crosses were aligned. He once again used the test cross to test the condition of the F1 heterozygotes. He knew that if he bred with a parent of known homozygous double recessive, he would get a ratio that was 1 to 1 to 1 to 1, as indicated here. So here's his test cross, and that only produces one type of gamete, and this is very important. We don't have 4 boxes here because there's no need for 4 boxes, because this individual only produces a single type of gamete, small r, small g. The individuals that he was testing, they produced 4 different types of gamete because they were double heterozygotes. And then this 1 by 4 Punnett square shows us the ratio of the expected phenotypes. In a Trihybrid-Cross, you guessed it, we're going to look at 3 different genes, 3 different traits. Each one will be independent of the other when it comes to the mathematics. The important thing is for you to keep each gene separate from the other. So we have 3 genes here, and each one is in the diploid condition, 2 chromosomes each. So this will be one parent, and this will be the other parent. And when they're crossed together, they will produce a triple heterozygous offspring in the F1. And that will then give us many more possibilities for when we breed these together with themselves. So the F2 will be pretty complex. The good news is that we can use mathematics to help us in easily predicting the categories of F1 gametes before producing the F2. And that formula is given here. It's 2 to the power of n, where 2 is the 2 alternative forms of allele at each gene, big A, small a, and n is the number of genes that we want to predict the gamete types for. In this case, 3. So putting that together, we end up with 2 to the power of 3, which is 2 times 2 times 2, and the answer is 8. So we should have 8 different types of gametes, not 4, when we go with a Trihybrid-Cross. So that will give us an 8 by 8 Punnett square. We can also use the forked line method by applying it to each gene individually to decipher the different types of gametes that can be produced. If you take the time to look at the process and then the results and look for a pattern, that's the best way to learn this. So I'll leave that to you. Or you may, like me, use the Punnett square method to get the same data. Let's continue and look at the middle part of the chapter, which talks about mathematics and probability. As we mentioned before, Mendel was trained in mathematics and he knew about probability, and he was able to make use of this tool. There are 4 rules of probability theory that we need to know right now, and they are the product rule, the sum rule, the conditional probability, and the binomial probability. The product rule is applied if 2 or more events are independent of one another. Then simply you calculate the likelihood of each, and then you multiply the 2 together. That is what we call the product rule, the multiplication rule. The sum rule is applied in another scenario, and it defines the joint probability of occurrence of any 2 or more mutually exclusive events. And in this case, you just add up the 2 individual probabilities. Conditional probability is the one that students sometimes struggle with. In this case, the outcome of something has already taken place, but based on additional information, we can now modify the probability calculation. An example always helps with this. So here's that example. If we cross a parent who's heterozygous with another parent who's heterozygous, the question is, what is the probability that the yellow-seeded progeny are heterozygous? Now, the yellow-seeded progeny make up 3/4 of the offspring, and they would include the big G. We're not interested in the homozygous recessive, small g, or the green-seeded progeny. So they would be then conditionally removed from the analysis. Now, because the yellow-seeded offspring are not GG, what's left over are just 3 boxes in the Punnett square. And 2 of those 3 boxes will be heterozygous, and the 3rd box will be homozygous. So if I remember, we did something similar to this in another slide, and if I can go back and pull that slide forward, that will help us. So here we're looking at not big G but big R, and not small g but small r, but the principle is the same. We want to know how many of these 3 boxes are going to be homozygous or heterozygous. But this one will never be part of the understanding because it has a completely different outcome. So we can remove this box and we're just left with 3, and the ratio between the 3 is the answer. Returning back to this slide, now we can answer the question, what is the probability that the yellow-seeded progeny are heterozygous? And the answer is 2/3. This is a conditional probability outcome. Not to worry, over the next few chapters, we'll get to practice this again and again until it becomes firm. Likewise, we'll get to practice binomial probability as well, but let's understand what it is first before we apply it. Binomial probability is applicable to situations in which 2 or more possible outcomes are possible each time. The approach expands the binomial expression, P plus Q equals 1, and that is then reflective of the number of outcome combinations that we are seeking. Oh my God. Okay, let's carry on and look at this. So let's start with the simplest example. So a binomial expression contains 2 variables at its core. That is P, the frequency of one outcome, and Q, the frequency of the alternative outcome. It's like tossing a coin, heads versus tails. So the chance of getting a head is 0.5 and the chance of getting a tail is 0.5. Add them together and you get 100%, which is 1. Same thing with the chance of getting a boy and the chance of getting a girl with one pregnancy is 50-50. So add them together, you get 1. This is great for predicting the outcome of a single coin toss. But suppose you had to perform 10 coin tosses and you wanted to see the likelihood of getting heads 10 times with a single coin. Then you have to expand the binomial expression to the power of n, where n is the number of successive events. How can we illustrate this? Let's go with this example. Suppose you wanted a family of 3 kids. What's the probability that you could have 3 girls? And this can be calculated using this expanded binomial expression. In fact, the answers can be more diverse. Here we can get all the different types of possibilities for boys versus girls. So the probability of getting a boy is half, 0.5, and the probability of getting a girl is again 0.5. So let that be represented by P and Q. Because we're asking about 3 children, we expand our binomial expression to the power of 3. And if you do that, you get this mathematical relationship. Using this and plugging in these numbers, we end up with the following outcomes. So P3, which is 3 boys, would be 1/8 of the time. So 1 in 8 chance of getting 3 boys. Getting 2 boys and 1 girl, you would have 3/8 of a chance. Getting the converse, you would also have 3/8 of a chance. That's 1 boy, 2 girls. And getting 3 girls will be down again to 1/8. So the total should add up to 1, and it does. But you can see, using this very simple formula, we can now utilize that as a genetic counselor, for instance, as we saw in another chapter, to predict the outcomes. Here is one other example to which this is applicable. Suppose you were interested in all the pea pods that contain 6 peas, and you wanted to know which proportions of seeds could be yellow, which proportions of seeds could be green in an individual that was heterozygous and self-fertilized. So pretty sophisticated analysis, but not really. Think about it. You're doing a self-cross with a heterozygous monohybrid cross, and you want to know for all the pods that contain 6 peas, what are the permutations of yellow to green. So you put in the probabilities. So P, the probability of a yellow pea from the Punnett square will be 3/4 of the time. And the probability of getting a green pea is 1 box out of the Punnett square, so 1/4 of the time. Using something called Pascal's triangle, which is available, you can then answer the question. So the number of events is 6 peas in a pod. And the question then becomes, what are the possible combinations you can get 2 colors of 6 peas in a pod? And the answer is 64. If one is visually inclined, then we can see how that calculation from Pascal's triangle would be calculated longhand. So here, there's only one possibility of getting 6 peas, all the same color of yellow. Likewise, there's only one possibility of all the peas being green, and they're arranged in a linear fashion. Here we can see 6 different ways in which you can have 5 green peas and 1 yellow pea. And likewise, the converse of that. And you go through all the possibilities, you end up with 64 possibilities. And you can go further and break it down into the binomial probability equation here. And this is P plus Q to the power of 6. And down the bottom, you can see the frequency of occurrence for this class is given right here, based on the frequency of the 2 outcomes. So P equals 3/4 and Q equals 1/4. A very convenient use of mathematics to aid genetics. Let's move on to the next thing, which is a statistical test used quite frequently in biological research called the chi-squared analysis test. It's basically a procedure to remove bias when your experimental results are on a borderline between being accepted or rejected based on a hypothesis. It relies on a comparison of what you expect to see and then what you do see. And that's it. What you do see is the observed, the O. What you expect to see is the expected, the E. And this is the basic formula. The chi-squared is the sum of what you observe minus what you expect squared divided by what you expect. And you do that for every observation. Once you work out the mathematics, you should get a single value for the chi-squared, which is called the probability value, the P. What's the probability that you're trying to calculate? That is best read as stated here. It is the probability that the results of another experiment of the same size and structure will deviate as much or more from the expected results by chance as yours did. So you're comparing your values to other potential experiments. Small chi-squared values are associated with a high p-value, and that simply means that your experimental data is very close to what you expect. There's not much deviation, which is good news. If, however, you got a high chi-squared value, that means then your data is way off target and there's some probability that your data is not accurately reflecting what you were testing. The actual p-value obtained is influenced, i.e. dependent, on something known as the degrees of freedom. The degrees of freedom is a pretty complicated parameter, but an easy way to understand it is it's equal to the number of outcome classes minus 1, i.e. the number of independent variables. By reference to standard chi-squared table values, we can then estimate the likelihood of accepting or rejecting the outcome. So we have columns here which refer to the degrees of freedom, and then we have probability estimates here with cutoffs, confidence levels, right? So this is the 5% confidence level. So if you convert that to a percentage, it becomes 5%. This is 1%, and this is 0.1%. This is 10%, and all the way up to 95%. So the cutoff in most scientific experiments is at the 5% level. Anything below 5%, your data is more variable than 95% of the time that other experiments will be very close to the acceptable data. So you're way out there in the Pluto zone in this region here. Here you're getting closer to the sun, and the higher you have a value here, the more likely that your experimental and your observed data are in agreement. Thus, for your degrees of freedom, let's say 5, what you want to do is have a chi-squared value which is over here on the lower side of 11.07. Then you can say that you are going to accept the hypothesis that your data and the expected data are real. Anything beyond 11.07, you will start rejecting your hypothesis and have the assumption that your data is way off and it doesn't represent what you were testing. The 5% cutoff value has been agreed to by mathematicians and others. Thus, in a simple sense, when any experimental result has less than a 5% probability, the hypothesis is rejected by convention. The chi-squared analysis of Mendel's data reveals a very, very strong correspondence between the predictions and the expectations. So all his data are supported by his theories. Let's take the results of one of his experiments and apply it to the chi-squared test. So in his notebook, he did this experiment where he bred round seeds with wrinkled seeds, a monohybrid cross. And he observed 5474 round seeds and 1850 wrinkled seeds for a total of 7,324 seeds. So this is what he got. This is the observed data. The expected data, which he calculated before he got the actual cross results, he predicted that 3/4 of all the seeds, these, should have been round. And he also predicted that 1/4 of all the seeds should have been wrinkled. So these are his expected values. So putting them into a chi-squared formula, we get this answer here. 0.263. That's the probability. And the number of degrees of freedom in his case is 2, the two categories, round versus wrinkled, minus 1. So the answer is 1. So looking at the values on the table for degrees of freedom along this row here, we just check the value. And the value that we have is 0.263, and that falls between 0.263 falls between here for 1 degree of freedom. So between 50% and 70%. So that's well above the 5% cutoff. So this data is in very good agreement with the expected. Therefore, his hypothesis that there's a 3-to-1 ratio is true, supported at least. Table 2.5 does a similar analysis, but this time on his Trihybrid-Cross data. And looking at all the numbers and the expected, and doing the chi-square value test, we come up with a probability value of greater than 90%, which is fantastic. Moving on now to the final part of Chapter 2, we begin to analyze different ways in which information from parents to offspring can be transmitted. Although we have some genes that are transmitted according to the patterns Mendel discovered, not all our genes are subject to those patterns. In fact, more than 2/3 of our genes are transmitted in a more sophisticated manner. We have 46 chromosomes in a somatic cell. Each chromosome has a partner, so there's actually 23 pairs of chromosomes in a diploid organism, such as humans. Those chromosomes are categorized into two categories, the autosomes and the sex chromosomes. So the autosomes have their own mechanism of inheritance, which is not exactly mirrored by the sex chromosomes. So it's important to deal with autosomal inheritance as opposed to sex chromosomal inheritance or mitochondrial inheritance. The one pair of sex chromosomes in humans is designated X and Y. Males have an X and a Y, females have two X's. Sometimes it's not possible to conduct experiments on organisms, as Mendel did with plants, without ethical and moral obligations. So performing experiments on humans comes under that umbrella. So what do? We rely on existing family architecture. And by laying out that architecture into generations and following the transmittance of alleles and chromosomes amongst family members, we come up with something called a pedigree. So a pedigree is a family tree that allows us to trace the inheritance of traits in humans. A typical pedigree will display, using standard notation, the relationship between members of that family. So the first thing to remember is that circles represent females and squares represent males. If you are subject to an unspecified sex, i.e. a miscarriage or a future pregnancy, then that's indicated by a diamond. A completed circle, a filled circle, means that that individual expresses the trait that's the subject of that pedigree. A line through a symbol indicates a person that's deceased. Other lines are used to connect different regions of the pedigree. So generations are represented by vertical lines, and horizontal lines represent parents. And if two parents are closely related genetically, we can also use a double line. Adoption is indicated by a dotted line. And any siblings for a couple are indicated by a bracket system where the firstborn child is always on the left and the last child is on the right. Identical twins, if they are maternal, are indicated by a line joining the two branches, whereas fraternal twins don't. They're like two independent children. And then numbers are used along the rows to indicate generations. And then within each row, Arabic numbers are used to indicate the order of the individuals. There are many more symbols used in pedigrees, but these are the common ones and the ones that we should be familiar with immediately. So let's go back to that autosomal mode of inheritance. So autosomal dominance is one mode of inheritance out of 6. So autosomal dominant traits follow a particular pattern, and they have 6 characteristics. And they include that males and females have the trait in equal frequency. If a child has the trait, then at least one parent had to have had the trait. Either gender can transmit the trait to their child, and if neither parent has the trait, then none of their children can have it, unless you're told that a rare mutation has occurred. If the traits are rare in the population, that means less than 1%, then those with the trait are likely to be heterozygous. And in matings or crosses where one parent has the trait and the other does not, approximately half the offspring will have the trait. If both parents have the trait, then they may produce children who don't have it. And the reason for that is that both parents are most likely heterozygous. Figure 217 shows a prototypical family pedigree where a trait has autosomal dominant pattern of transmittance. So we can see this mother, she's deceased, so is the father. And that's generation 1. That'll be individual 1, generation 1, individual 2. Then we have generation 2 with individuals going right across. Once the pedigree is complete, we number it. And we do this likewise with generation 3 and generation 4. Lines are used to connect the family members, and people marrying into the family come in on their own, and then they are associated with the partner, and the number of children they have is indicated by lines coming down from the main fork. So in this case, this couple had a son followed by another son who's deceased, followed by a daughter, and then another son. Autosomal recessive inheritance offers us 6 clues to its presence. First thing is, males and females have the trait in approximately equal numbers. Individuals who have the trait are often born to parents who do not have the trait. The reasoning behind this is that both parents are heterozygous. Now, if both parents have the trait, then all the children born to them will have the trait. The trait can skip generations. It may appear in the grandparents, but not the parents, and reappear in the grandchildren. Or some children may have it and other children may not. Again, in conditions where the trait is rare, less than 1% in the population, and only one parent has the trait, a child can only have the trait if the other parent is heterozygous. Figure 218 represents a pedigree of an autosomal recessive trait. We can see here that the trait is passing from unaffected parents through to their children, but not every child gets it. So this is typical of an autosomal recessive condition. We'll have more to say about inheritance in other chapters. To conclude, let's go back and see if we can marry up what Mendel discovered with current modern molecular biology and genetics. We now know that alleles and their transmission is equated directly with DNA. And DNA molecules can have different information, and those variable DNA sequences are the alleles. And they act through messenger RNA and in many cases produce proteins, and those proteins are responsible for the phenotypes. So the first molecular analysis to be performed on Mendel's initial traits was published in 1990. This is fascinating stuff. If you recall earlier in this chapter, we spoke a lot about the shape of the seed. Well, recently we've discovered a gene called SBE1, and that produces an enzyme in the pea plants, and that converts the starch molecule amylose to a complex branched form of myelopectin. The presence of this enzyme is enough, even in a heterozygous format, to convert most of the amylose to a myelopectin. So seeds homozygous for the mutant allele, they lack the enzyme completely, and thus they contain amylose and they don't contain any myelopectin. So what are the consequences of this difference in this sugar? The answer is that amylose attracts water, and that water is lost as the seed matures, leading to the wrinkled appearance. So having too much of this produces a wrinkled appearance. Having too much of this does not. In the next case, we can tally the length of the plants with another gene called LE, or Li. Now, Li produces a hormone that's very important in plant growth. That hormone is called gibberellin, and its production requires the presence of an enzyme that's encoded by the LE gene. So plants with a dominant allele can produce the hormone and grow tall, but all it takes is one copy. You don't need two. But plants that are homozygous recessive, they don't produce the enzyme, and therefore they have very, very low levels of the hormone, and therefore they are short in phenotype. Another example is the seed color, yellow seeds versus green seeds, and that's controlled by another gene, SGR. So in yellow peas, one or more alleles of the functional SGR gene produce an enzyme, and that's important for the breakdown of chlorophyll. So that initially green seeds lose chlorophyll and then they turn yellow as they mature. We've discovered further that the mutation prevents production of a functional enzyme. Seeds that are homozygous for the mutation are green due to the absence of the enzyme's activity in chlorophyll breakdown. One more finding has to do with the color of the flowers, white versus purple, and this is due to a discovered gene called BHLH. So the product of this gene is not an enzyme, it's a protein. It's a transcription factor, and that transcription factor activates the expression of certain genes, including the genes responsible for producing the pigment anthocyanin. All you need is one dominant allele to be able to produce the transcription factor, and that produces the anthocyanin. Plant cells that are homozygous for the mutant don't have this transcription factor, and therefore they cannot activate the genes responsible for producing this pigment, and they stay white, the default color. Table 2.8 summarizes all these findings in an easy-to-relate format. In the case of Mendel's traits, it appears that each one was a loss of function mutation. That means a mutant allele does not produce something that's needed to produce the wild-type appearance of phenotype. All it takes is just one functional dominant copy to override the mutation. So if you have a heterozygous condition, then the normal copy, the capital letter, is enough to produce enough biochemical product to overcome the mutation. If, however, you have two broken alleles, neither one of them is functioning, then you have total loss of function, and then that biochemical pathway will be non-functional. The loss of function mutations must be homozygous, therefore. Loss of function is a very common utilization when it comes to different phenotypes. We'll see this in multiple chapters. In concluding this chapter, we can now make the following correlations: that the inheritance of alleles precisely parallels the pattern of transmission of phenotypic variants, proven by molecular biology. In addition, the phenotypic variation that results from differences in structure and function of proteins is responsible, in most cases, for allelic function. So alleles alter the structure and function of proteins, and they can then generate either defective pathways or alternative pathways. Molecular analysis has led to the identification of DNA sequence differences between alleles. So we know now that there's a base, at minimum, different between one allele and another, and it could be more profound than that. And then, of course, functional analysis of protein products for each allele has led to the understanding of its role in producing the phenotype, and that understanding is increasing every day. Thank you so much.